Welcome back to the Deep Dive. Today, we are taking a look at something really foundational for the entire bioindustrial sector.
We are.
We're focusing on how these brilliant scientific discoveries, things like next-gen biomaterials, these complex nutritional ingredients, how they actually move from a lab bench to a real commercial product, something you can actually buy. The science is often just phenomenal, but we've seen so many smart startups get stuck. They get stuck right at that chasm of scale.
Scaling trap. It's very real.
Exactly. So here's the really painful question we're digging into. How do bioindustrial founders scale up their amazing processes without, immediately bankrupting themselves building these incredibly expensive custom pilot plants?
It is the ultimate trap. You need continuous industrial data to convince investors to give you that big check.
Right.
But you need the big check first to build the facility that gives you the data.
So it's a catch-22.
Completely. But the sources we've looked at show a really clear answer emerging. And it's a fundamental shift in what we think of as shared infrastructure. We're seeing CROs, contract research and development organizations, and especially CDMOs, the manufacturing ones, stepping way outside their traditional farmer roles. They're becoming the essential shared infrastructure, the utilities, you could almost say, for the entire consumer biomanufacturing industry.
So if I'm a founder, my first thought is, why wouldn't I just spend a bit more money and own my own production line? Have total control. What's the real advantage here?
It all comes down to the CAPEX advantage, capital expenditure.
Okay.
I mean, think about what a good CDMO already has. Not just the massive, expensive steel tanks, but decades of operational know-how.
Right, the expertise.
Exactly. The playbook we're seeing now suggests that the smartest founders, they don't even try to build pilot plants from scratch anymore. They build relationships with CDMOs that have already solved the hardest scale-up problems for platforms like theirs.
I remember talking to a founder once who said building their first pilot line felt less like launching a company and more like sinking a very, very expensive anchor.
That's the perfect analogy. And that is exactly the feeling you're trying to avoid.
So the market is backing this up.
Oh, absolutely. The global CDMO market is already huge, well over $250 billion. And it's growing at, around 9% a year, pretty reliably.
And what's driving that?
It's the complexity. These new biologics for consumer applications, food ingredients, probiotics, sustainable materials, they're just demanding more specialized, shared resources.
Let's put a number on this for you, the listener. If you're thinking about building your own pilot line, say a 1,000-liter system with all the downstream purification stuff, that's a 9-figure commitment over a few years.
Often before you have a single confirmed customer.
Exactly. What we're seeing is that outsourcing that initial phase can cut your early capital needs by anywhere from 40 to 60%.
40 to 60. It's a staggering reduction in risk. And all that cash you saved.
It goes back into the science.
It goes back into what makes your company unique. Perfecting the biology, optimizing your strain, and this is the big one, paying for customer validation and getting those early commercial agreements signed, proving the market is there.
Okay, so it sounds like a perfect solution. You reduce risk, you save cash, you scale faster. But there's always a but. Outsourcing must bring its own trade-offs.
Of course. It's not a silver bullet.
What are the new risks a founder is taking on with this model?
That's the crucial question. You're basically exchanging a huge upfront capital risk for ongoing operational and execution risk.
Okay.
And 3 big risks really stand out in the data. First one is capacity bottlenecks.
Meaning everyone wants the same thing at the same time.
Precisely. If everyone in the sector raises a Series A in the same quarter, suddenly the best CDMOs are booked solid for a year. And your whole scaling timeline just evaporates.
And your market window could close.
It could.
Yeah.
Second is pricing power asymmetry. A small startup desperately needs that capacity. And the big established CDMOs, they know this.
So they can squeeze you.
They can absolutely dictate terms. Squeeze your margins, lock you into unfavorable contracts. It happens.
That makes the word partner sound a bit dangerous.
Yeah.
How do you protect your core IP in that situation?
And that's the third and honestly the most critical risk. IP and data. Sharing a facility means you need to be incredibly diligent.
Right.
You need rock-solid, upfront contracting around your processes, your analytics, and especially the unique strain data that is your company's crown jewels. You have to draw really clear lines.
So what they learn on their equipment versus what I brought in the door.
Exactly. Who owns the process optimization data versus who owns the core biological IP? It has to be crystal clear from day one.
So the guidance is clear.
Mhm.
Treat them like a strategic partner, not a vendor. Build a real relationship.
You have to.
Okay, let's move inside the facility then. We've got the why, the capital advantage. Let's break down the what. What are these specialized CDMOs actually delivering? It's not just renting a big steel tank, is it?
Oh, far from it. They're providing these end-to-end capabilities that bridge that huge gap between a cool lab result and a product you can actually sell. We can really break it down into 3 core layers.
Let's do it.
Okay, so layer 1 is the foundational technical work, upstream and downstream development. This is where the real process optimization happens. They take your lab strain and they iterate on it, usually at that critical 10 to 100 liter scale.
And what's the biggest value add there?
De-risking the physics. They run continuous fermentation, run specifically to de-risk the physics of scaling up.
Ah, the physics. That's where things always break, right? What works in a flask doesn't work in a giant tank.
Never. In the lab, everything is perfect. But in a 10,000-liter vessel, nonlinear effects just take over. We're talking about critical things like Ka, the mass transfer coefficient.
How you get oxygen to the cells.
Exactly. In a flask, it's easy. In a massive tank, your cells can literally starve of oxygen even if you're pumping it in.
Yeah.
So the CDMOs, they have the equipment and the know-how to manage all that, the mixing, the heat transfer, all the hard stuff.
So they solve the scaling problems. And then once you have your fermented liquid, your broth, what about getting the good stuff out?
That's the downstream part. They nail the purification, which is often way more complicated than the fermentation itself. They're focused on consistent recovery yields.
How much you actually get out.
Right. And for consumer products, this is non-negotiable. Establishing a clean impurity profile that meets all the safety and quality standards.
Okay, so that's layer 1. What's layer 2?
Layer 2 is the quality and regulatory infrastructure. Now, this might sound a little dry, but this is the invisible barrier to entry that saves startups years of work and millions of dollars.
It's the boring stuff that can kill a company.
It is absolutely mission critical. If you want to sell a food ingredient or a cosmetic, you need validated quality systems. CDMOs provide that instantly.
So things like GMP.
Things like GMP, HACCP for food safety, ISO 22000. They handle the long, tedious stability programs, the analytical method transfers, the batch record systems you need for traceability.
And what about navigating the regulatory maze, like for new materials?
They provide that support too, helping founders put together, say, a GRAS dossier, generally recognized as safe for a food ingredient, or a food contact notification for a new biomaterial. If you skip this layer, you don't have a product. You just have a science experiment with no path to market.
Got it. And the third layer gets us even closer to the actual customer.
Right. This is the application-specific services, and this is what proves your material actually works in the real world.
Okay, so what does that look like?
What's really fascinating here is how close they get to the final SKU on the shelf. This layer includes formulation development, stability testing, but in real product matrices.
So not just testing the powder on its own.
No. You're testing your new protein in a low pH carbonated beverage to see if it crashes out, or making sure your probiotic can actually survive in a complex cosmetic cream.
No.
And for biomaterials, they handle pilot converting trials for packaging. This means running your new film on actual industrial equipment, things like form-fill seal lines, FFS lines.
The high-speed machines that big brands use.
The very same. Because if your amazing new material tiers or jams on a real FFS line, it doesn't matter how great your biology is, it's commercially useless.
That really drives home how end-to-end this partnership has to be. Okay, let's shift to the funding playbook, because this whole model just completely rewrites how founders should be raising capital.
It really does. There's a huge difference between funding the idea and funding the execution.
Right. Seed funding is for the idea. Series A is for execution.
Exactly. Seed equals biology plus your first TEA, your first economic model. Series A equals pilot physics plus real customer validation. And there are no shortcuts.
Okay, let's break down the seed stage. We're talking $500,000 to $2 million, maybe a 6 to 12-month runway. What does a team have to deliver?
The goal is twofold. Prove the core science works, and prove that it's at least plausible that it could be economical. The deliverables are super specific. You have to generate continuous, reliable data on your titers, your yields, your productivities in that 10 to 50 liter range, not just one perfect batch. Consistency. You have to build that techno-economic analysis, the TEA, and it needs to show a minimum selling price, an MSP, that's within, say, 1.5 to 2 times the cost of whatever you're replacing.
And using realistic assumptions.
Using realistic assumptions, no magic. And finally, you have to get that first customer letter of intent or a joint development agreement, some proof that someone actually wants to buy this thing.
That TEA is the real gatekeeper. Show us the path to competitive pricing, or it's a no-go. Okay, so then you get to Series A, the execution round, 5 to 15 million. What's the new goal?
Series A is all about de-risking the scale-up physics and getting deep into application integration. That money has to cover 3 to 6 months of continuous pilot runs in the big tanks, the 1,000 to 10,000 liter range. You're proving commercial stability.
And what's the single make-or-break metric for those big runs?
Yield consistency. You have to prove that your yield variance is less than 10%, even when you're using worst-case feedstocks.
Because in the real world, your feedstock quality is going to swing all over the place.
It absolutely will. And if your yield collapses when that happens, your whole business model collapses. That variability is where companies bleed money.
So what are the investors looking for on the financial side at Series A?
They're looking for much more sophisticated modeling. You need to deliver a risk-adjusted net present value, an NPV, that clears at least a 20% hurdle rate.
So proving the project is fundamentally profitable, even with all the risks baked in.
Exactly. And you need to show with sensitivity analysis that even if your feedstock costs jump or the market shifts, the project is still a solid investment.
Wow. That is a whole other level of rigor. Okay, let's quickly touch on a tool for managing all this complexity. I know we've been seeing this concept, the Bioindustrial Readiness Radar.
Yes, it's a great way to visualize risk beyond just the science. TRL, technology readiness level, only tells you if the science works in a lab. The radar forces a much more holistic view.
So what's on it? How does it work?
You can picture a spiderweb chart with 5 axes, each scored 1 to 10.
Okay, what are the 5?
First is technology and application, so TRL plus ARL, application readiness. Second, manufacturability, your pilot costs, your yield consistency. Third, feedstock robustness, how well you tolerate variability. Fourth is regulatory maturity. And fifth, crucially, is market traction, your offtakes, your JDAs, your pilot orders.
That's powerful because 2 companies could have the same TRL, like a TRL6.
But have wildly different radar shapes. You could have Platform A with amazing biology, a 9 on technology, but a 3 on feedstock, and a 1 on market traction.
While Platform B has solid biology, maybe a 7, but they've proven they can handle feedstock variability, a 9, and have strong customer validation, an 8.
Right. An investor looking at that radar for Platform A immediately sees the problem. They need to stop tinkering with the strain and go get some customer agreements.
Okay.
It forces everyone to confront the balanced risk picture.
Let's wrap up with a really practical playbook then. For the technical leaders listening, what are the, say, 3 non-negotiable rules for actually getting this done?
Number 1, design backward from the SIGU. From the final product on the shelf. You have to co-develop prototypes on real industrial equipment with your partners.
Not in the lab.
Not in the lab. Run your films on real FFS lines. Test your ingredients in real beverage formulations with full processing like pasteurization. You have to simulate the real commercial environment or you'll fail on day 1 of launch.
Okay, that's number 1. What's 2?
Build commercially literate technical teams. Every single senior scientist and engineer has to be able to answer the so what questions.
The business questions.
The business questions. How does this 10% titer improvement actually change our minimum selling price at 10,000 tons a year? They have to understand the cost implications of every technical choice.
And the final piece of the playbook.
Choose defensible, high-value niches. Go after segments where you have a real barrier that protects you from copycats.
Like what?
It could be high-performance materials that need a proprietary coating process, or nutraceuticals that have strong clinical data and complex patented formulations, or maybe an integrated biorefinery that pulls 3 or 4 valuable products from one single feedstock. You need complexity that creates a real economic moat.
So that focus on predictable execution using these strategic partners, that's really what separates the science projects from the actual sustainable companies.
That's the whole shift. It's moving from being just about the biology to mastering this integrated, predictable execution, all driven by these CDMO relationships and very rigorous metric-driven funding stages.
So here's the final thought for you to take away from all this. The next wave in biomaterials and bioprocessing. It won't be won by the team with the best story, or even the team with the best strain. It will be won by the teams that can turn all that messiness, the feedstock variability, the scale-up physics, the regulatory complexity into predictable, investable execution.
That's the new benchmark for success.
It is. Thank you for joining us on this deep dive.
Thank you.
We'll catch you next time.